as we get. Box in the part where it says laws of probability, different color highlighter from what we did, chi-squared stuff. We probably outlined the bottom box. We probably outlined metric prefixes. So grab you a different color highlighter and highlight and box in laws of probability. So the important takeaway that I want to make sure, and I want you to add this to your formula sheet, is whenever we are trying to find this or this, so we have two options, and it's either this option or that option. We can choose either one. When it's or, we are going to add. So or means to add. So I can either have this characteristic or this characteristic, and that means I'm going to add. If I need this characteristic and this one, I need both of them at the same time. It's not a choice. I need them both. If it's and, then you multiply. So and equals multiply. If it's an or situation, you add. If it's and, then you multiply. All right, then you have a little sheet that looks like this. Grab that. And I want you to take it and I want you to fold it in so it creates two doors that meet in the middle. So fold it in so that you have two doors that meet in the middle. Before we get to that point, so I've got my little doors and they're folded closed, and so I'm writing stuff on the outside. P1 generation. Does that sound familiar? P1 generation? Let's see if this helps. Better?
Yes.
Okay. Figuring things out as we go. So, P generation I have on the left-hand side, and on the right-hand side I've got capital T crossed with lowercase t. And if you need to move up, or if it's too dark, you can move around, do whatever you need to do. Hang on, I gotta grab this real quick. So P generation, parental generation for us, and in all intents and purposes, how we're gonna use that, it's gonna be pure breeding generation. So pure breeding generation means you have one individual that's pure for this characteristic, the other individual that's pure for a different characteristic. So that's why the capital T crossed with lowercase t. Homozygous dominant, purebred individual. Homozygous recessive, purebred individual. That means for the homozygous dominant, capital T, throughout that individual's entire family, there's nothing but capital T's. No lowercase t in there anywhere. It's pure breeding individual. For lowercase t, a homozygous recessive individual, entire family is nothing but recessive alleles. Pure breeding individual. With me so far? Okay. So, P generation. Next is the F1 generation. So, for the F1 generation, F1 on the left-hand door, to get the F1 generation, I have to create a Punnett square that crosses my P1 parents. So notice how I use color to show where everything's coming from. Parents are always on the outside of the Punnett square, one allele in each spot. You fill in the inside of the Punnett square for the individuals that are the F1 generation. So now I'm going to take two F1 individuals and I'm going to cross them. What do you think that generation is called? F2, second allele generation. Look at how brilliant you are. So now I have my F2 generation, which means I have to take two F1 individuals and cross them. Parents always go on the outside. Offspring always go on the inside. So my F2 generation, I've highlighted that orange. The yellow on the outside represents the parents, which are F1. The orange on the inside represents F2. So now go to the inside, open it back up. All right, let's just look at the top first of all. So the top on the left-hand side, that's the type of cross that we're looking at. Across the top, that's a monohybrid cross. What does mono mean? One. So monohybrid, that means we're only looking at one characteristic at a time. P generation for us is always going to be pure breeding. So it's capital T crossed with lowercase t, lowercase t. So if that got cut off on yours, that should say P generation equals homozygous dominant crossed with homozygous recessive. Then, our examples of what we can cross. So as soon as we cross those guys, we can have our F1 generation, F2 generation. Let's say we have different parents that we're starting with. We can have heterozygous parents. We can have homozygous dominant crossed with heterozygous, homozygous recessive crossed with heterozygous. So those are examples of P1 generation, or of following generations that we can use to cross. Notice that they're written in different colored ink, right? So this, I want to say I wrote in pencil, and this, both of these are in blue ink. Does that make sense? Do you see that on your paper? There's a reason for that. They're written that way specifically. The ink color that they are written in corresponds to their genotypic ratio. What is a genotype? Genotype. The expressed part is the phenotype. What we see, what we observe is phenotype. So what's genotype? Sort of. Pardon? Sort of. Genotype is you're thinking genes. You're thinking too hard on the first day back. Genotype represents the genes. So alleles, kind of, sort of, capital, lowercase, what kind of options do they have? But the genotypic ratio represents what are the genes that we're going to get out of that cross. So if I have capital T, lowercase t crossed with capital T, lowercase t, then my genotypic ratio is going to be a 1 to 2 to 1 ratio. So that means I'm going to have one of this kind of genotype, and I'm going to have two of this genotype, and I'm going to have one of that genotype. Think about it for just a minute. Think the little Punnett square in your head. If I put that little Punnett square in my head and I put capital T, lowercase t, capital T, lowercase t, one of the offspring are going to be what genotype? Capital. Two of them are going to be capital, lowercase, and one's going to be kind of like, oops, I don't have that on the front here. Kind of like what we had right here. So I have a 1 to 2 to 1 ratio. Does that make sense? See where those are coming from? All right, so then you're going to trust me on all the rest of them because we're not going to run through all of this. This is just stuff that you need to know, and this will be a resource that you come back and use whenever they're asking for a ratio, whether it be genotypic or phenotypic. You just come back and use this. For the love of Pete, use the tool because you use it often enough and you're going to memorize it anyway. So both of these right here are in blue ink. So if I cross those, the ratio we're going to have is 1 to 1. How many offspring do we have in a Punnett square? 4. So how is my ratio 1 to 1? 2 are going to be the same, and then the other 2 are going to be the same. And then it's simplified. So 2 are going to have this genotype, and the other 2 are going to have that genotype, and we're just simplifying it to have that ratio. With me so far? Thumbs up, thumbs down, somewhere in the middle. Okie doke. All right. Underneath, it says phenotypic ratio. What did we say phenotype was? What's expressed, what we observe, what you see, hear, smell, taste, touch, characteristics that we're able to observe. Phenotypic ratio a lot of times is probably, I mean, you'll use genotypic ratio, but I really think that phenotypic ratio is going to be a little bit more important to us, at least at the beginning, because that's what we can see. We don't know what the genes are. You'd have to run tests and pull DNA and run gel electrophoresis to find genes unless they give it to you. But phenotype, whoops, boop, you look at it, and I can see if it's red flowers or white flowers or tall or short. I can see the phenotype, so that's helpful. All right, so grab two different color highlighters. Nope, 3 different color highlighters. Oh, let me do that. All right, so let's look at this real quick first, just so you can see where it's coming from. So let's say capital T is tall, lowercase t is short. For this generation right here, for my F2 generation, how many of them are going to be tall?
Like, 2 T's is tall, you said?
Capital T is tall.
Capital T.
So 3 of them.
And how many are going to be short?
1.
Okay, so this one's going to be tall because it has at least 1 dominant allele. This one's going to be tall, at least 1 dominant allele. This one's going to be tall, at least 1 dominant allele. And this one's going to be short. So what's the phenotypic ratio?
3 to 1.
3 to 1. Make sense? So for this one right here, I've underlined it with a pink highlighter for me personally. You can use whatever color you want, doesn't matter. And then I matched it up with its phenotypic ratio. Then I did the same thing for the others, and so you're just going to have to trust me on those. But if you want to do it and take the time, you can go back and run those through. So this one, underlined pink, and then I highlighted 3 to 1 because that's going to be the phenotypic ratio every time. It doesn't matter what the letters are. If I have 2 monohybrid crosses with their heterozygous, the letters are always going to, no matter what the letters are, it's always going to be that phenotypic ratio. If I cross homozygous dominant with heterozygous, they're all going to have the same phenotype. Does that make sense? Can you picture that? Imagine that Punnett square. If I cross these two, every single square is going to have at least 1 capital T in it, so they're all going to be tall. If I cross a heterozygous with a homozygous recessive, my ratio, phenotypic ratio, is going to be 1 to 1. How many offspring are usually in a Punnett square?
4.
4, so why do I have 1 to 1?
Simplified. 2 of each kind.
So it's simplified because there'll be 2 of these phenotypes and 2 of those phenotypes, and that's where I get the 1 to 1 ratio. So this is pink, pink, orange, orange, yellow, 1 to 1 yellow. Okay? All right, so next is dihybrid crosses. Bottom part is dihybrid cross. Once again, what we started with was pure breeding individuals. Homozygous dominant, everything's capital. Cross with homozygous recessive, everything's lowercase. Pure breeding individuals, that would be the P generation. For the F1 generation, F2 generation, F3 generation, we can have so many different, and this isn't even all of them. I just put a few down there so that we would have options, and we can take a look at it. So notice how the ink color is different. I can't even. So I've got the first 2 and the last one. Those are all blue ink. Then this one right here is pencil, and then this one here is red ink. What do the ink colors that things are written in, what do they correspond to? The genotypic ratio. So whenever I were to cross this one, or this one, it's going to have a 1 to 1 to 1 to 1 genotypic ratio. Do you remember how many boxes are in a dihybrid cross? 16. So why do I have 1 to 1 to 1 to 1? Simplified, same amount of everything, 4, 4, 4, 4, because that would add up to 16, so it's simplified. I cross this one right here. I have a genotypic ratio of 1 to 1 to 2 to 2 to 1 to 1. I cross these 2 right here. I'm going to have a genotypic ratio of 1 to 2 to 2 to 1 to 4 to 2 to 1 to 2 to 1. I'm going to tell you right now that most common genotypic ratio, the one that you're going to see a lot, is the 1 to 1 to 1 to 1. You might see those, but not as often. Don't run the 16 square Punnett square. Come over here. Look to see what individuals you're crossing and what genotypic ratio it is. There's absolutely no need to do any more work than that. Okay, highlighters. 1, 2, 3, 4, 5 different colors. just out of curiosity, for this first one right here, any idea what kind of results we're going to get if we cross a homozygous dominant with heterozygous individual? Okay. What about, and we're looking at phenotype, so let's not worry about genotype right now, but phenotype, what we will see, what we will observe.
They'll all be tall, whatever.
B is, whatever capital B is. Because of the fact that I can't ever come up with two recessive alleles, just looking at that, bam, I know when I cross those, I should expect to see every offspring having the dominant traits. Do you see how I can say that? I can't possibly come up with a recessive trait. This parent has to give an allele, this parent has to give an allele. If I don't have any recessive alleles to give, then I can't end up with anything recessive. So that's an easy one. So I've highlighted that one green, and then all the same phenotype green because those match up. The second cross, capital, lowercase, capital, lowercase, lowercase, capital, lowercase, that one I've highlighted blue, and that's going to be a 1-to-1 ratio. You don't need to think anything about that. You see those, if you don't see, there's two easy ones on here. If you don't see those two easy ones, just come to this foldable and look up what the ratio is. The third one, I have capital, lowercase, capital, and all heterozygous. That's going to be a 3-to-1 ratio. The yellow one is another easy one. Not really easy, but it's going to be one that you see a lot and should be burned into your brain. Anytime that you see that they are crossing two heterozygous individuals, that should automatically tell you that it's going to be a 9-to-3 to 3-to-1 ratio. Automatically. If I see that I'm crossing two heterozygous individuals, bam, 9-to-3 to 3-to-1. That should be automatic. So these are the two easy ones. If you see homozygous dominant crossed with heterozygous, or actually crossed probably with anything, you'll never get a recessive allele if it's homozygous dominant, then I know that they're all going to be the same phenotype. Bam, that should be automatic to you. So that should be all the same phenotype, which is this on the bottom. Then if I see two heterozygous individuals, bam, that's 9-to-3 to 3-to-1 phenotypic ratio. Then if you see any of these others that don't come across real easily, use on any of them at all. In fact, use the foldable, come back, see what ratios we've got those matched up with. Okay, so far? Okay. Grab the worksheet. When we look at multi-hybrid crosses, it is still related to everything that we did with the monohybrid cross. It's just all put together. So you can kind of think of it being pieced apart whenever you're doing multiple characteristics. Remember, if it's the same characteristic, you're using the same letters. So if I say, if I have B's here, capital B, let's say, is blue, lowercase b is red. But that's the same characteristic, that's flower color. It's the same characteristic. The T's have to do with height. Capital T tall, lowercase t short. So it's the alleles, it's the two different options from the same characteristic that we're talking about, the same gene but different alleles. Capital T, lowercase t, same letters because they both have to do with height. Capital B, lowercase b, two different letters, but, well, same letter because they both have to do with flower color. So I can talk about height and flower color at the same time. I can use T's and B's at the same time, but they're talking about two different characteristics. That's where we get that dihybrid crosses from. When we look at multi-hybrid crosses, we can look at each characteristic individually whenever they independently assort. And for right now, if you get further on into biology and genetics and stuff like that, they don't all always independently assort. They don't move independent of each other. Some genes are linked, but for us, we're just going to assume that they're all on different chromosomes and they can independently assort and go into gametes however they want to go into gametes and however meiosis puts them in there. So I cross these two individuals, I get a Punnett square that looks like that. I cross these two individuals, I get a Punnett square that looks like that. But, like I said, we can look at both of those characteristics at the same time. So let's say I'm crossing this individual and that individual. So if I cross this individual and that individual, if you were to write out the 16 square Punnett squares, first you've got to figure out what gametes you're going to use, then you have to put the gametes on the Punnett square, then you have to set up the Punnett square, then you have to fill in the Punnett square. It's very time consuming. And there's lots of places where you can make a mistake, because if you just fill stuff in wrong, then you'll make a mistake here and make a mistake there, whatever. So everything I've seen, nobody's asking AP Bio students to write out the 16 square Punnett square just to come up with an answer. If I cross these two individuals, what's the probability of getting X offspring? But I do want to show you where that comes up with. So I can find these gametes and these gametes from this one. That's where they're going on the Punnett square. So these all go across the top, these all go across the side. I can fill in that entire Punnett square, and I would fill it in the same way. Everything on the left-hand side goes into each box on the right. Everything on the top goes into each box below it. You would do it the exact same way. It's just really time-consuming. So the one on your worksheet is probably a little easier to see. You can see that it's already been filled in for you. And so here would be the question that they would ask. What is the probability that the parents, capital T, lowercase t, capital B, crossed with heterozygous for both traits, will have a capital T, lowercase t, capital B offspring? So these parents are crossed, and we're looking for this offspring. So I've already gone through there, and I've circled the 4 possible. There's only 4 possible that have that exact offspring on there. So there's 4 out of 16 total. That equals 1/4. So that's the probability, 25%, 1/4. We're going to keep with fractions, and then when you get at the end, just know that you might have to go between fractions, decimals, percentages. They can give you the answer or ask for the answer in any one of those forms. But if you get to 1/4, you can convert that to 25%, right? Or 0.25. Good with that? All right. So flip it over to the back. Let's do this in a slightly easier way. Same question. What's the probability, oopsies, that these parents, crossing these parents, are going to end up with this offspring? We can look at each characteristic individually. Because we're looking at stuff that independently assorts, we're good. So we can look at them independently. So I can go ahead and do a Punnett square for height and a Punnett square for flower color. Once I've got my Punnett square for height and my Punnett square for flower color, I just use the laws of probability. I need offspring that is capital T, lowercase t, and capital B. I need both of them at the same time. It's an and. It's not I need this or this. I need those both at the same time. And means I'm going to multiply. And means multiply. I need that and that, and that, so I'm going to have to multiply. All right, so we need offspring that has capital T, lowercase t, and capital B, so that means I need to multiply. So first, what's the probability of capital T, lowercase t with these individuals? If I go back up there to that Punnett square, oopsies. Right, but let's not, don't reduce yet. I promise you it's going to help. It's going to be easier because if we're adding and you reduce and this is 1/2, but this is 1/4, well then if you add them together, now you've got to make the denominators the same and you have to do, so let's keep it simple and not reduce until you get at the end. So, right. So In this Punnett square right here, I want capital T, lowercase t. So this is capital T, lowercase t. This is capital T, lowercase t. So 2 out of 4. So the probability of capital lowercase is 2 out of 4. I also need capital B, capital B. I go up here to this Punnett square. I've got 2 out of 4 capital B, capital B. I need to multiply because I need both of those. So capital T, lowercase t, 2 out of 4 times capital B, 2 out of 4 equals 4 out of 16, and then you can reduce. It'll be easier, maybe not necessarily in this one, but there will be problems that it's going to be easier just to wait to reduce at the very end. Does that make sense so far? Questions so far? Okay. These two are going to eventually go into your AP Bio Spiral notebook. Grab the little worksheet, the little half worksheet. And in fact, go ahead and take out your spiral notebook. Let's take and put the foldable. I've got it on the left-hand side page because I've got my worksheet on the right-hand side page. So I've got the foldable, and I've taped it to the top, and then I've given myself some definitions underneath, so I know what monohybrid and dihybrid crosses, what those terms mean. So I've taped the foldable on top on the left-hand side. And underneath, I've got monohybrid cross. It's a cross involving only one characteristic, for example, height, tall or short. Dihybrid cross, the cross involving two characteristics. So now we've got flower color and height. So this sheet of paper is taped in. This worksheet's taped in on the next page. Alright, so a little half sheet of paper is taped all the way up on the top, and then a little bit of information underneath. So take a look real quick at each one of these problems. These represent the parents, and this is what they're asking you to find out what the probability is. So this is, we want to look, what's the probability of this offspring? So these are the parents, what's the probability of offspring? These are the parents, what's the probability of offspring? These are the parents, what's the probability of offspring? Do me a favor real quick in two different color highlighters, just to point out the fact that this is one set of parents, so highlight that A color. And then notice that every other set of parents on this page are the exact same set of parents that we're using. So this is a different set of parents than every other problem on this page. So this, highlight this one color, then choose a different color and highlight those the same. So I highlighted this one orange on mine, and I highlighted these three blue on mine. It is important to pay attention. What are the parents that you're crossing to find out what the probability of the offspring is? Then underneath two sentences, the fractions are for the offspring. 1/4, 2/4, 3/4, 4/4. The fractions determine offspring, determine the probability of the offspring. So fractions, F reminds me offspring. Punnett squares, you need to use the parent genotypes. The Punnett squares use the parent genotypes. The fractions are offspring. Fractions determine offspring. Punnett squares are using the parent genotypes. fractions determine offspring. Punnett squares use the parent genotypes. All right, let's look at the first problem. Give me this. All right, the first problem, and you're going to see it better on yours, so just look at yours. I've got capital D, capital S, lowercase s crossed with heterozygous for both characteristics. It wants to know the probability of a roly-poly that is dark-shelled with 4 stripes. We do need to take a step back. Oh, okay, let's do this real quick. Capital D is dark. Capital S is 1 stripe. Lowercase s, and remember, find a way to make sure that you can tell the difference between your capital S and lowercase s.
Is lowercase d light?
Light, yeah. Mm-hmm. That would probably be helpful, huh? Whenever you start with one of the problems, you can make it easy on yourself to start. Okay, so I want to know dark-shelled 4 stripes. What is a genotype for dark-shelled? Need a little more.
It'd be capital D, capital D, or capital D lowercase d.
All right, so capital D, or capital D lowercase d. Both of those would give me dark shells, correct? So before I go any further, I want to see if with these parents, those are possible. If I have this parent and this parent, would it be possible to have offspring that's capital D lowercase d?
Yes.
Yes. Would it be possible to have capital D, capital D?
Yes.
So it's possible for both of those. All right. 4 stripes. What do I need to have 4 stripes? Can I have capital S lowercase s? This is the only genotype that's going to get me 4 stripes, correct? With these parents, is it possible to have a lowercase s, lowercase s?
Yes. Wait, yes,
Do you see why?
yeah.
I can get 1 lowercase s from one parent, 1 from the other, so it is possible. If any of these are not possible, so let's say I'd have capital, if that's not possible, I would just mark it out. I would put an X through it and not even think about it. But the first thing I would do on any problem is look to see what genotypes am I even looking for in this situation. So the thing is that I can have an offspring that is capital D, lowercase s, and it will have dark stripes, or it will have dark shells and 4 stripes, right? Or I can have an individual that's capital D, lowercase d, capital S, and it can have dark stripe or dark shells and 4 stripes, right? So I can have this or this and still get that offspring. Does that make sense? All right. Your spiral notebook. I have this parent, capital D, lowercase d. We're going to look at dark shell. We're going to look at shell color. And then we're going to look at striping differently. We're going to separate them out. So this parent has heterozygous on both S's, homozygous dominant, heterozygous on the D's. So underneath, at the bottom of your spiral notebook, fractions are offspring, parents are Punnett squares. So I put 2 little Punnett squares. Do you see how these represent the parent, the D's from the parents, and the S's from the parents? So go ahead and do those Punnett squares real quick and fill them in. Don't worry about that third one just yet. Go ahead and do those 2 Punnett squares and fill them in real quick on the bottom of your spiral notebook underneath that little worksheet. Okay, so I'm going back up to the worksheet. I'm gonna put it on here just so it makes it a little bit easier to see. But this is where I'm at. I have this problem and I'm gonna write right underneath there so I can see exactly where things are coming from. So I just wrote it a little bit bigger and a little bit darker over here. That's it. Got it?
Yep.
Okay. All right, so I'm right here underneath that on that worksheet, and I'm going to put everything that I need underneath so we can see where it's coming from. So look at your Punnett square. Your Punnett square for shell color. What's the probability that I would end up with capital D, capital D? Let's put it in fractions and not simplify. 2/4. And we don't want to simplify just yet. We're going to keep it nice and simple for ourselves. What's the probability on the other Punnett square that I'll end up with lowercase s, lowercase s? 1 out of 4. What's the probability that I'm going to end up with capital D, lowercase d? Lowercase s, lowercase s. All right, so first of all, do you see where I get those fractions from? Parents, Punnett squares. Once you fill the Punnett squares in, fractions. Now I'm trying to find out what's the probability of these offspring. Okay, this says I can have this one or this one. Or means I'm going to, if it's this offspring right here, I need to have this and this at the same time. So what am I going to do with these two fractions? What am I going to do with these two fractions? Okay, order of operations. Can I just go straight across? What do I have to do first? So I need to multiply, get those answers, and then add them. And so for right now, we're still going to keep things simple without simplifying. What's 2/4 times 1/4? Do you see where I get 2/16 from? When you multiply the numerators across, multiply the denominators across. And so what's going to be in here? 2/16. And so what is my answer? And then we can simplify now and get, how are we doing so far? The reason why we're not simplifying, so let's say this is 2/16 and this is 1/16. You have to go back and get common denominators to get it to 16, to add it together to get your answer. So don't reduce until at the very end, because your answer would be 3/16 and you wouldn't reduce that any further. Okay? All right. Let's look at the next one. The next one has different parents. So on the second one, I don't have capital D on the first parent. Both individuals are heterozygous. So at the bottom of your spiral notebook, you're going to need one more Punnett square, right? You're going to need a Punnett square like this so that you can, and then I mean, you can rewrite Punnett squares every time if you want, but why?
Question, on the last one, I just noticed this, but it said like dark shell with 4 stripes, and you only need like a capital D for it to be dark shell, and on the Punnett square that like we filled out, they were all like capital D around, so would that be still 2/4 or would it be 4/4?
So there are cheats. I'm showing you baby steps, and then I've had several students come up with cheats that, so far as I can tell, work out the exact same way. And I don't have enough math in me to know why, blah, 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 whatever. So everything that they have in common when they come up with the cheats is that they keep on doing and multiply or add. So there are other ways to solve the problem that might be even easier for you. I don't care. For some people, they have to set it up this way. I don't care. So long as what you're doing is following the laws and you get the answer that's a correct answer and you're using the right stuff, I don't care. But sometimes it's like algebra. Sometimes, you know, you have to see it in a different way, and people see things in a different way. I need to see what you do. I need to see your work. Because if you come up with an incorrect answer, I need to help you find out why. So no matter how you get to that answer, show me how you get to that answer, and I don't care if it's the right answer and if it applies the laws. Does that make sense?
Yeah.
So there are workarounds to it, absolutely. The first way that I did it, I always did it where I pulled the offspring out and had to look at it that way. That was the only way I understood it. And then when a student showed me and walked me through how they did it, I was like, oh, well, that's so much easier. But for me, it always clicks one way first. I just need to see what you're doing. The reader needs to see how you get that answer. Because if you just throw something out there, you're not proving that you know what you know. So just show what you're coming up with. Okay, so next problem, dark-shelled roly-poly. Okay, dark-shelled roly-poly, what's the possible genotype?
Capital D, lowercase d, capital D, capital D, capital D.
Okay, so if I look at these two parents, is it possible to get capital D from them? Is it possible to get capital D, lowercase d? Okay, 4 stripes. What's my genotype? Okay. If I look at these parents, is it possible to get homozygous recessive?
Yes.
Okay, so this looks exactly the same as this. What's the difference?
There's a homozygous dominant.
The parents are different. So you're using different Punnett squares. That's the only difference for this problem. Now in the next problem, it'll be the same parents and they're going to be asking for different offspring. Right? So for these guys right here, that means if we're doing it the baby step way, I could have an individual, or I'm looking for an individual that is either capital D, lowercase s, or one that is capital D, lowercase s, lowercase s. And I'm going to put that on a big sheet of paper. Do you see where I get that part from? So underneath, baby steps, because you're going to show Mrs. Johnson where you're getting stuff from. Remember, we have to use the appropriate Punnett squares for the parents. You cannot use the Punnett square. I don't know where my sprung notebook went to. You are not using this Punnett square because these are not the parents. We're using this Punnett square because those are the parents. All right, so on the appropriate Punnett square, what is the probability that I'm going to get capital D? 1 out of 4. What's the probability that I'm going to get lowercase s? On the appropriate Punnett squares, what's the probability I'm going to get capital D, lowercase d? Lowercase s. Okay? So this says or. Or means I need this and this. So what am I going to do with those fractions? Multiply. And. I need this and this. So what am I going to do? Can I do straight across? What's order of operations? What do I have to do first? I've got to multiply first before I add. What's 1/4 times 1/4? 1/16. 2/4 times 1/4? 2/16. What's my total if I add those together? Perfect example of why we don't reduce until at the end, because it's just easier that way. All right, I want you to do those other two problems, and then I'm going to take a look at them eventually.